Assalamu alaikum. How are you doing? I hope you're doing fine. Let me introduce myself. I am Dr. Ma'moon Ahram from the School of Medicine. I'll be covering genetics and genetic disorders and modes of inheritance related to dental and oral conditions. I have three lectures. And they will be covered into or divided into three sections. During the first part, I'll cover introduction into molecular biology, genetics, and few techniques, as well as chromosomal anomalies. The second part, I'll discuss Mendelian inheritance and Mendelian diseases. And then in the last part, I'll cover the multifactorial diseases and conditions. So the main resource for you is this lecture itself and whatever I say. You can refer to Emery's Elements of Medical Genetics, very nice book. I'll provide you with that textbook. You can refer to it. It's a resource. If there's something that you would like to elaborate on, if you'd like to get more information on any topic, you can refer to it. It's a very nice textbook. I also encourage you to visit the online Mendelian Inheritance in Man website, a nice and informative website that discusses or it gives you information in regards to different diseases and conditions in man. So you can just type in the name of the disease, syndrome, condition, whatever, and you will get a lot of information in regards to that, as well as if you type in, for example, a name of a certain gene, it will give you also information on diseases related or associated with this gene. So let's start. Just to introduce what molecular biology is. Well, a long time ago, maybe 50 years ago, scientists came up with what is known as the central dogma of molecular biology, which basically what it tells us is that DNA can be replicated by a DNA polymerase enzyme. It can also be used as a template to be transcribed, producing RNA, a process known as transcription, and this is done by an enzyme known as RNA polymerase. And then RNA can be used or it can be translated using ribosomes, tRNA, whatever transfer RNA, producing proteins, and proteins do the functional part in cells. Now that was 50 years ago, the central dogma of molecular biology. It still exists, of course, but then in addition to these processes, we do have more details, or we have more information, and there are additions to that model. So basically, the RNA itself can be reverse transcribed. So that's reverse transcription by an enzyme known as reverse transcriptase, where you can have DNA formed. This is true for retroviruses like the HIV virus, the HIV virus mainly. Well, also the RNA itself can be replicated by enzymes known as the RNA-dependent RNA polymerases. So they're different than these because these are DNA-dependent. These are RNA-dependent, so RNA itself can be replicated. A great example is the coronavirus that is making a huge mess globally. So it does have an RNA polymerase that is used to replicate its RNA. So that's the central dogma of molecular biology. Now, DNA is composed of four bases. The A, G, C, T, all are deoxyribonucleotides. They are ordered in certain ways. In our genome, if we compare two human individuals, if you compare the DNA sequence of two human individuals, you would find that they are similar by 99.9%. So we have a difference of 0.1%. These DNA is double-stranded. It's a helical molecule, and the two strands are associated with each other via hydrogen bonds. Now, it can be organized with proteins known as histones. It's wrapped up, forming this large structure known as a chromatin. A chromatin means it's a molecule that is made of both both DNA and protein. Well, eventually, its DNA is packed into a large genetic unit known as a chromosome. Okay. Now, certain regions within the DNA contain a code that represents genes, and these genes are the ones that are transcribed and then translated. So there's a starting point for any gene and an end point where you have termination, start and termination of transcription, and either one of the two strands can be used. Okay, so for this gene here, that's the beginning, that's the end. For that gene, that's the beginning, that's the end, and so on. So every gene has a certain code or order or sequence of nucleotides. Now, we do have the same exact genes, and we have these genes located on the same positions in our chromosomes. And again, we are similar by 99.9% in general. Okay, so when translated, they are translated according to a codon. So we have this codon table. It shows how they are translated from these codons into amino acids and eventually proteins. What ribosomes do is that they read with tRNA, they read three nucleotides, that's a codon, and they are translated into amino acids again. Okay, and this is what the genetic codon is all about. So I'm going to go quick on that just to refresh your memory on what we're talking about. So transcription or gene expression in general can be regulated at different levels. So we have transcriptional control, that is the rate of transcription, the start and stop of transcription can be regulated. We have a processing of the messenger RNA by alternative splicing, that's true in human cells, producing different messenger RNAs. Mature processed messenger RNA can then be transported, and this can be regulated, and then it can be translated. Or it can be degraded. Okay, so again, that's another level of regulation. Then you have production of a protein, and this protein again can be regulated by different mechanisms like chemical modification, phosphorylation, for example, or it can be degraded and so on. Okay, so there are different levels of regulation of gene expression. What we mean by gene expression is basically production of protein starting with DNA. Okay, so the main level, the predominant level of regulation is transcriptional regulation. Okay, so basically every cell has the same chromosomatic cells. They have same chromosomes, exactly identical chromosomes, but we have different genes that are expressed. So in our genome, we have about 20,000 genes, but not all of them are expressed in every single cell. Rather, you can have certain cells like muscle cells, for example, they can express 6,000 genes. You can have skin cells expressing 5,000 genes. Some of these genes are common, like actin, for example. Others are cell-specific, so you can have skin-specific cells, you can have skin-specific genes, you can have liver-specific genes that are transcribed, and so on. Okay, so and this is controlled by transcription factors overall. So for example, in skin cells, you can have a gene that is transcribed at moderate or low levels, because you have the core proteins that bind to the promoter region upstream of a gene. inducing transcription. In liver cells, you can have very high expression of the same exact gene because you have other regulatory proteins that aid or facilitate or activate or speed up the process of transcription. Okay? The same gene may not be transcribed at all, or it can be transcribed at very low level in brain cells, for example, because you have an inhibitor molecule, a repressor that binds also at a different site on the DNA, suppressing or reducing transcription. So this is basically transcriptional control. Another level of regulation at the transcriptional level is very exciting, and it's making, although it has been known for a long time, but now it has made great strides in terms of medicine, whereby the DNA itself can be modified chemically. Okay, and this is known as epigenetic control, and this is very exciting. So basically, you can have methylation of cytosines in what is known as islands, CPG islands, that is regions that are rich with C's and G's. And when these cytosines are methylated, then you have wrapping up or packaging the DNA so that it cannot be accessed by transcription factors. That basically causes inhibition of gene expression. So you can have methylation of the DNA. You can also have acetylation or methylation of the histones that wrap up DNA. So if they are acetylated or methylated, you can have activation of transcription, because now the DNA is relaxed, genes can be accessed by transcription factors and so on. If they are, if acetylation is removed, then you have packaging of the DNA. Okay, you can have also phosphorylation of histones, and this phosphorylation can suppress transcription. And then we have something new, and it's very, very exciting time in molecular biology. You have non-coding RNA molecules, that is RNA molecules that are transcribed but not translated. And an example is long non-coding RNA molecules, and what they do is that they can bind to DNA, allowing the interaction of activators, transcription factors, to DNA, inducing or repressing transcription. Okay, so that's overall, this is what epigenetic regulation of DNA, of gene expression is. Okay. Okay, now DNA can be mutated, and that would cause different diseases. And there are different types of mutating DNA. So you can have what is known as you can have silent mutation or silent variation of DNA. So for example, we have these codons in a gene, and they encode these amino acids. So AUG would encode for methionine, GCC would encode for alanine, and so on. Okay, if you have a single base pair mutations, okay, let's see here. So if you have a single base pair mutation whereby C is altered to T, that would not change the amino acid. It would still be alanine. So this is a silent mutation. Now you can have what is known as nonsense mutation. Now in this case, you have a mutation of C to A, and that would cause a termination of translation. Okay, so this is called nonsense. يعني عندك sense يعني منطق, nonsense إنه ما في منطق, يعني there is nothing that can be read. Okay, so actually if you go back, and you don't have to memorize this, of course. So if you go back to the termination sequences, that's in RNA, UAA, UAG, UGA. So if you go here, okay, again, you look at the sequence, TGC, so it is, if it becomes TGA, that is UGA, this is a stop codon. That's termination of translation. Okay, you can have another point mutation is known as missense mutation, and in this case that would cause a change from T to G, that would cause a change of the amino acid from cysteine to arginine. Okay, so this is called missense, meaning انه منطق غلط مش مظبوط. طيب, you can have mutations that are most of these mutations, single point mutations, because there's a change in one base, they are the most common type of mutations in DNA. Okay, now sometimes you can have addition or deletion of a base. So right here you can have deletion of a base that would change the frame of reading of codons. So instead of having, for example, by deleting this base, which is C in this case, the codon would actually be GCT. Okay, that would result in addition of an amino acid known as alanine, and then that would change everything that comes afterwards. So frame shift mutations, either by deletion or insertion, would change everything that comes after that mutation. So right here you have addition of a C. So now this C would be part of the next codon. Now instead of reading TGC, you would have you would have CTG right here, and then that would change everything that comes afterwards. Okay, so so we have again different types of mutations. We have substitution mutations where we change the base. That would be silent, missense, or nonsense mutations. Now, synonymous mutations are silent mutations, so meaning that there is no change in the amino acid sequence of the produced peptide. Non-synonymous mutations are either missense or nonsense mutations, because non-synonymous would mean that you have an altered amino acid, a different amino acid that is produced. Okay, you can have mutations in splice site promoter enhancer in regions other than the coding sequence. So we're not going to go into details of that. You can have deletion. If you have deletion of multiple of threes, and so if you have deletion of this codon, for example, right here. So if you have deletion of this codon right here, that would mean that you will have deletion of a cysteine. Now, but if you have deletion of, let's say, this C followed by AA, that would again cause a frame shift mutation. Okay, so multiple of threes, that would cause deletion of amino acid. Not multiple of threes, that would cause a frame shift mutation. Sometimes you can have large deletion, that is, you can have deletion of several codons. Again, it's the same exact thing. It can cause deletion of amino acids or frame shift mutations. Same thing with insertions. Okay, now, in molecular biology, these mutations can be represented in certain codes or nomenclature. So for example, if I have a missense mutation, which is basically change of an amino acid to another, okay, so we would say in terms of nucleotides, C would represent cDNA, that is, which is basically a DNA that is made from RNA. Okay, by reverse transcription. Okay, so if I say C 4A2G to A, that would mean that at position 4A2 of the cDNA or the messenger RNA, we are changing G to A. That would cause a change of the amino acid within the protein sequence, which is arginine at position 117, that is amino acid number 117, which was arginine. Now it changes to histidine. Okay, nonsense would, in terms of the position of nucleotides, it's a change of nucleotide number 1756, G to T, and that would cause a change of glycine at position 542 in the peptide sequence to an X that is to a stop codon. Splicing is indicated by a plus sign. Deletion of one base pair, this is 1078, deletion of a T at position 1078. That would change a frame shift mutation, represented right here with FS, frame shift, okay? Changing valine to tyrosine and everything that comes afterwards, okay? This is deletion of 3 base pairs, deletion of CTT, for example, from position 1652 to 1654. That would change deletion of an amino acid that is phenylalanine, and this is in frame. Now, if we say that there is insertion of a T, that would cause again a frame shift mutation, again FS, starting with a change of leucine to phenylalanine, and this is again a frame shift mutation. Okay, now let's talk about a technique that is making a huge change in the field of genetics and medicine. So basically, there are different techniques that we can talk about, but I'm going to talk about only one of them, and that is next generation sequencing. What we mean by sequencing is basically knowing the order of nucleotides or the sequence of a DNA, the order of nucleotides within a gene or a piece of DNA. So this is what sequencing is. Now, in the old days, sequencing was done using a substrate known as dideoxynucleotide. But although it has been really beneficial and we still use it, it's very slow and it's not good. It's not useful for doing large-scale sequencing of human genome. So the Human Genome Project started in 1989 with the cooperation of 7 countries and multiple centers, and what they wanted to do was to sequence the human DNA, that is knowing the order of nucleotides in the human genome. Okay, and it took them 15 years to finish the project, and it cost about $3 billion. So that's a lot of money and a lot of time. Now, in 2007, there has been the introduction of several techniques for DNA sequencing, and they have collectively been known as next generation sequencing techniques, where basically nowadays, by the way, you can sequence a whole genome, that is the whole DNA of a cell of an individual, in about 24 hours instead of 15 years, and it would cost about $500–700 instead of $3 billion. So there has been huge improvement in the technology. Now, there are different techniques for next generation sequencing, but this is one of the most famous ones and most reliable and used techniques. So basically, the way it works now, since we have the database of the human genome, the sequence of the human genome, well, we can integrate both the technology itself with the informatics or with the DNA analysis. So basically, we take a human DNA and it's fragmented into different pieces. Now, notice that some of these pieces can overlap with each other. Again, but we're not talking about taking a single DNA molecule. We're talking about taking a lot of the of DNA molecules from the same individual. Okay, so when this DNA is fragmented, it's fragmented into smaller pieces. Some of these pieces overlap with each other, and then you have the addition of adapters. Okay, these adapters are added to the ends, and the sequences of these adapters are known. Then the DNA fragments are bound or attached to a solid surface. Okay, and the DNA itself is amplified, meaning that in the same cluster right here, you would have several DNA fragments on the same spot. So starting with one, you would have multiple. Now it's shown in here too, but in reality it's a lot of DNA fragments attached to the same cluster. Okay, so basically, and then each one of them can be sequenced. Okay, and it's sequenced using nucleotides that are special. They have two important characteristics. The first one is that when a nucleotide is added, the polymerase that synthesizes or amplifies the DNA cannot add the next one. Okay? So right here, when a G is added, the next one cannot be added. So that's one, until it's activated. Okay? Meaning until it's chemically modified, whereby the polymerase can add the following nucleotide. Okay, that is complementary to the next nucleotide in here in DNA. That's one. So that's the first feature. The second feature is that once it's activated, it emits a light, and this light can be detected. Okay, so there are two things. One, when the first one is added, or when a nucleotide is added, the DNA polymerase cannot add the following nucleotide until the nucleotide that is added is activated. And the second one is that once it's activated, it emits a light, and this light can be detected. Okay? So you can imagine that we have the solid surface, and this happens all at the same time. DNA fragments with different sequences. The DNA polymerase would add, or polymerases again, there are multiple enzymes. They add a nucleotide to each one of them, and then they're activated at the same time, and each one of them would emit a certain light that is detected. And then the cycle is repeated. That is, now you can have another one that is added. Okay? Again, it has to be activated, it emits a light, and the second cycle you have detection of light from each one of these clusters. And the technology allows for detection of the light that is emitted from each nucleotide. What's important as well is that each one of these four nucleotides would emit a certain light. So if this emits, let's say, red, it would be read as, the computer would read it as, let's say, a T. If this emits a green color, then it means that the nucleotide that is added to this DNA fragment is an A, let's say, and so on. So, and the computer can read all of these lights that are coming out from the solid surface, all at the same time. So we would know the sequence of each one of these fragments, and then they are collected, and they are, since we have the sequence of the whole DNA, these fragments are placed together into one complete sequence. So, so that's the idea. And here, each one of them would emit a light. So we add a C, there's a light that is emitted, then it's a T at this position, then another T, then A, then G. So, and this is read by a camera. It takes pictures of all of these lights that are coming out. Now notice that each spot gives a certain color. So it's almost like actually fireworks, and that's how it looks like. In this little space, and it's only 100 microns, okay, we have all of these spots. Now we're talking about a surface that is about the knuckle of a finger, the size of a knuckle of a finger. So we're talking about billions, actually, billions of lights that are coming out all at the same time, and they all can be read, by the way. So let me start this. So let me start this video for you. That's the company that has made this technology.
Sample preparation begins with extracted and purified DNA. The first step in Nextera sample preparation is tagmentation. During tagmentation, transposomes simultaneously fragment and tag the input DNA with adapters. Once the adapters have been ligated, reduced cycle amplification adds additional motifs, such as
Don't worry
the
about
sequencing
details,
primer
okay?
binding sites, indices, and preparation begins with extracted and purified DNA. The first step in Nextera sample preparation is tagmentation. During tagmentation, transposons simultaneously fragment and tag the input DNA with adapters. Once the adapters have been ligated, reduced cycle amplification adds additional motifs, such as the sequencing primer binding sites, indices, and regions that are complementary to the flow cell oligos. Clustering is a process wherein each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, 4 fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determine the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated, similar to the first read. After completion of the index read, the read product is washed off and the 3' end of the template is deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Index 2 read product is washed off at the completion of this step. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends blocked. The original forward strand is cleaved off and washed away, leaving the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is washed away. This entire process generates billions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments.
So you can refer to this video on the YouTube or this slide to understand it better. Don't worry about the details, just the information that I gave you in the previous slide. Okay, so what sequencing allows us, or the information that we can get from massively parallel sequencing is really amazing. So we can sequence a whole human genome. We can identify mutations, changes in the human DNA, deletions, insertions, you name it. So we can get a lot of information. So if we compare DNA of a person with a certain disease and we don't know what the genetic cause of that disease is, by comparing his genome to the database that we have, we can identify the mutations, and there would be a lot of mutations eventually by minimizing or filtering the information. We can tell what the exact gene that causes the disease is. So now we talked about DNA and a technology related to sequencing. So let's talk about the chromosomes. Now we are, remember something, we are diploid. So we have, meaning that we have 2 copies of every single chromosome. One comes from the father, and the other comes from the mother. Okay, so all of our somatic cells are diploid. The germline cells, sperm and egg, are haploid because there's 1 copy of each one of these chromosomes. Now, so we have maternal and paternal DNA, and before cell division, we can have replication of each one of them. So these 2 would be called sister chromatids. Each one of them is known as a chromatid, and these are sister chromatids. So here, these 2, okay, so these 2 are known as homologous chromosomes, the maternal and paternal chromosomes, like chromosome 1, for example, the chromosome 2, chromosome 3, etc. So these are homologous chromosomes, and when each one of them is replicated, now we have sister chromatids. Okay, now, if you look at the structure of the chromosome, you would notice that you have a centromere that sort of like it divides the chromosome into 2 regions, and these regions can be considered, one of them is considered a short arm, and the other one is known as the long arm. The short arm is known as P. Okay, so it is it has a symbol of P. P means petite, that is, small, short, and the other one is Q, that is the long arm of a chromosome. And they're separated again by a centromere. Okay. Now, chromosomes can have actually different shapes. You have metacentric chromosomes, where you have the centromeres almost in the center of the chromosomes. We have the submetacentric, that is, well, you clearly have a short arm and long arm. You have acrocentric, that is, you have the short arm really short, and you have and the long arm is really long, and you have what is known as telocentric, and telo means end. So whereby you have the centromere almost close to the end of the chromosome, so you don't have a short arm of a chromosome, you don't have a P arm of a chromosome. Something else about chromosomes that is important is that the ends of chromosomes are known as telomeres, and telomeres are very important in stabilizing chromosomes. So as we age, telomeres becomes shorter and shorter, and chromosomes become unstable. That results in cell death, and eventually that leads to death of a person as a result of aging. So this is aging. Okay. Now, there are different ways, techniques by which we can analyze chromosomes. One of the most famous ways is karyotyping, and in karyotyping what we do is that we stain chromosomes using a stain known as Gemza. So when chromosomes are stained, you have the formation of a structure that looks like this, where you have banding patterns. where you have dark bands and light bands within the chromosome. What these bands indicate is that how condensed the chromosome is, how packed the DNA is in the chromosome. So if the DNA is quite packed, it gives a dark stain. If it is loose and relaxed, it gives a light stain. Now, what's important here is that each chromosome has a banding pattern. Not only that each chromosome has a certain length, it does have a banding pattern that is unique to that chromosome. And when chromosomes are separated or isolated, they're isolated from dividing T cells in the metaphase. That is when the chromosomes are lined up before separation into the ends of a cell and before cell division. Okay, so this is one type, and it has been really useful and still used in laboratories. So another technique is known as fluorescent in situ hybridization. In this case, we use probes that are labeled. They are fluorescent, and these probes, they target specific sequences that are complementary to the probes, and the probes would hybridize or bind to chromosomes that contain sequences that are complementary to the probe, and you have fluorescence that comes out of that chromosome. Now, so you can have gene-specific probes. You can have centromeric probes. Centromeres have specific sequences that are also chromosome-specific, so we can label chromosomes at their own centromeres. We can have telomeric probes that label the ends of chromosomes, or we can use what is known as chromosome painting. That is, we paint the whole chromosome with so the whole chromosome is colored. Okay. Another technique that is, and this is still used in medical laboratories. Another technique is known as comparative genomic hybridization. Now, this solves a problem in karyotyping, in that is, we have to isolate chromosomes from dividing T cells, so the chromosomes have to be condensed, and it's really hard to do that when we want to analyze cancer cells and DNA of cancer cells, which are characterized by large deletions, or large abnormalities. So comparative genomic hybridization solves this problem, and it has been improved quite a bit using array-based CGH, or comparative genomic hybridization, whereby we have arrays that would allow us to compare large number of chromosomal regions within cells. So basically the idea in here is that we take DNA from a tumor cell, for example. It doesn't have to be in the metaphase again, and we have a reference DNA, a whole genome again. And this reference DNA is like from normal cells, like let's say T cells from normal individual. We label the whole tumor DNA, we label the whole reference DNA, and then we allow them to hybridize to each other, that is, anneal to each other. And if there is no deletion or duplication of a certain DNA region, there should be a 1-to-1 ratio. Okay, but if there is a gain of a certain color, like a green color in here, it means that you have duplication. If you have red color, it means that there is deletion of that chromosome because you have more red than green. As I said, this has been improved using arrays, whereby we have different chromosomal regions. Again, we label the reference DNA with normal DNA with a certain color, and the tumor DNA or the test DNA with another color. We allow them to hybridize and then bind to sequences that are placed on the surface area, and again it can be, the DNA can be read on each one of these spots. Okay, so these are the techniques. So let's go over chromosomal anomalies. But before, so this with the comparative genomic hybridization, the whole purpose is to see if there are any abnormalities in a whole chromosome in terms of duplications or deletions of a certain chromosome. Okay. So let's go over chromosomal anomalies. These can be divided into two types. We can have numerical anomalies. What we mean by anomalies is abnormalities in the chromosomes. So we can have numerical abnormalities of chromosomes, and there are different types of abnormalities. We can have something known as aneuploidy. With aneuploidy, it means that we have more than or less than 46 chromosomes. Now each cell normally would have 46 chromosomes. Now, if we have 47 chromosomes or 45 chromosomes, well, this is abnormal. So we can have monosomy, meaning that we have like monosomy X. It means that we only have one X in a female rather than XX. Okay, so in monosomy we have a condition, monosomy X. It's known as Turner syndrome. In this case, this individual would have 45 chromosomes rather than 46 chromosomes. Or we can have trisomy, like trisomy 21, that's in Down syndrome. Okay, or we can have that, which is chromosome 21. There are three chromosomes 21 rather than two. We can have trisomies of the sex chromosomes like XXX female or XYY or XXY. These are males. Now, XXY male is also, it's a condition known as Klinefelter syndrome. Or we can have polyploidy, okay, which is different. In aneuploidy, then we have an extra chromosome or a chromosome that is missing. But with polyploidy, we have, instead of having two sets of chromosomes, that is 46, we have three sets. So we would have 69 chromosomes, or 3n. So n is 23, okay? So instead of having 2n, that is 46, we have 3 n's, or we can have a tetraploidy, that is 92 chromosomes instead of 46 again. And this happens in cancer cells, okay? You can have these abnormalities mainly in cancer cells, but it can exist in fetuses, but they cannot survive, of course. Or we can have abnormalities in chromosomes that are related to structures. So you can have a part of a chromosome that is deleted. Now, it's different than the molecular deletion that we talked about before. In molecular deletion, we're talking about deletion that cannot be observed by karyotyping or the FISH, the fluorescent in situ hybridization techniques, or even the CGS. Okay. Rather here, with these chromosomal anomalies, we're talking about large chunk of a chromosome is deleted, and this can be observed by these techniques that I just mentioned. Or you can have insertion. Okay, you can have insertion of a part of a chromosome. You can have what is known as inversion. So in this case, we have inversion of a DNA, so it is flipped in here. Okay, you can have translocation, that is part of chromosome can be translocated. It can it can transfer from one chromosome to another. Okay, so you can have or you can have exchange, reciprocal translocation, that is, you can have part of a chromosome jumping to another chromosome, and the other chromosome would have its part jumped or exchanged for the other chromosome. Okay, there are different structural abnormalities as well. We can have what is known as ring chromosomes. In this case, you have a chromosome that is cleaved at the ends, and then both ends of the same chromosome would fuse with each other, forming ring chromosomes that look like this. Okay, So you can have different types of chromosomes that can form ring structures, like ring chromosome 20, which is associated with epilepsy. You can have ring chromosome 14 or 13, and they are associated with mental retardation as well as dysmorphic facial features. So throughout the lectures, I'll try to focus on syndromes and abnormalities, diseases that are related to dental and oral conditions. You can have ring chromosome 15, which is associated with mental retardation as well as dwarfism and microcephaly. So you can have anyhow. So this here, right here, is the example of abnormalities related to ring chromosome 14 and ring chromosome 13. Okay. You can have another structural abnormality of chromosome known as isochromosomes. Now notice in this case what happens is that you have the formation of two different chromosomal regions that are from the same chromosome, whereby you have a chromosome that has two P arms and another one that has two Q arms. So this is most commonly found in chromosome. That is in the formation of isochromosome X, and it accounts for like 15% of all cases of Turner syndrome. And patients with Turner syndrome can have abnormalities, craniofacial abnormalities. As you can see here, you can have the cross bites are lateral rather than what you call them, like straight bites. Okay. There are other chromosomal abnormalities like mosaicism, الفسيفساء. So basically, you can have a mutation that takes place during the embryonic development. So if you have a mutation in one of these cells, you can have cells that you can have the mutation distributed in different regions of the body. Okay, so this is what mosaicism is. Okay, and this can happen at the chromosomal or molecular levels, meaning that you can have a mutation, a point mutation, for example, a small deletion in DNA, or you can have a large chromosomal abnormality in terms of structure or number, but again, it would be differently distributed in the human body. Now, this is really important right here. This table is important, and I want you to pay attention to this table. So these are different terms that can describe karyotyping. So again, P is the short arm, Q is the long arm, cent means centromere, del is deletion. So for example, that's the nomenclature. So you can, with karyotyping, you can say 46XX del 1 Q21. It means that this individual has 46 chromosomes, so that's normal. This person is a female, XX, but she has deletion in chromosome 1, the Q21 arm of that chromosome. Or you can have duplication. So again, 46 chromosomes. The person is male, but there's a duplication in chromosome 13, region Q14. Okay, so we're talking about specific region on the Q arm, on the long arm of a chromosome. Fragile site, we'll talk about the fragile X syndrome, and so fra means there's a fragile site or a region of chromosome that is broken. Isochromosome, which I just talked about it, so we can say 46X, so that's female with a missing X chromosome that is actually an iso chromosome. Now inversion, so right here we have 46XX, that's female with 46 chromosomes, but there's inversion in chromosome 9 between the regions P, the short arm 12, and Q12, the long arm 12. So basically, in cytogenetics, the long arm and the short arm are divided into regions from 1 to whatever. So we know where this inversion takes place. In cytohybridization, that's in case we do the FISH analysis or the fluorescent in situ hybridization. If there is the abbreviation of R, it means that this is a ring chromosome, so 46XX female with 46 chromosomes, but chromosome 21 is not normal, it has a ring structure. T indicates translocation. 46XY, that's a male with 46 chromosomes, but there's translocation between chromosomes, or exchange, that is, between chromosomes 2 and 4, region Q21 of chromosome 2 with Q21 of chromosome 4. Okay, terminal or end tip of an arm, there is mutation in the terminus of a chromosome, whether it is P or Q. Mosaicism, that's for example male XY 46 chromosomes in some cells, but other cells have XXY and 40 chromosomes. Okay, so you need to know these nomenclature. I might provide you with a certain karyotyping condition, and you have to interpret what this means, or I can ask you the opposite, that is, I can describe what the abnormality is, and I ask you to write the nomenclature of that individual.